Hello, hello.
Yes, we can hear you. We can hear you.
Yes. Should I start now, or is it like 7 minutes before the lecture we start, or?
Oh yeah, I forgot we have a break. Sorry. Yes, we have a break. Good job. I think somebody's paying attention to the schedule. Okay, so we are going back at 10:40. We are coming back at 10:40 here. We will start that on time. So we can take a little break. I had my schedule collapsed. Do you want to share your screen?
Yeah, I will try. Dun, dun, dun. Is it okay?
Yes, perfect.
Okay.
Good. So I don't know if you remember from last year, but we had a little quiz afterwards. And
yeah. This year you didn't send me an email that I should come up with questions. So,
oh,
but
right.
now
So we kept it from last year. So we have this little quiz.
Yeah. Okay. I don't know what questions there was last year. I'm only going to talk about one parameter anyway.
So you will discover the quiz together with everybody else and see if it makes sense at all.
How many students are at the course?
20.
Oh, good.
Perfect. I think we have been only 1 year when we actually didn't fill the course. Last year we had 19. Otherwise, it's always full. But, oh, are you frozen? You look frozen.
I look frozen.
Yeah, oh, now you're not frozen.
Yeah, maybe I'm not having a perfect network. I'm actually sitting in Uppsala.
All right.
And running over Udrome because I'm going to go to a microscope demo at
right. Okay. Yeah.
12:30.
It sounds okay. Just a bit frozen for a little while.
Okay.
Yeah, 20 students, and we have most of them this year. I think we have 9 who are in Stockholm or Sweden. I think no, we have 9 in Stockholm, 2 in Linköping, and the rest are all outside.
Yeah, I just told people here in Uppsala that they should take the course.
Okay, good. They're welcome.
Maybe you get students next year.
Yeah. That would be nice. It's quite common that we have students from Uppsala as well. But this year we have no one from KTH, I think.
Yeah, maybe when I start presenting, I should turn off my video so the bandwidth is enough.
Yeah, and no problem. I mean, it works. It seems to be working quite nicely. I can tell you if you want, if something goes wrong. We want to see your face, Hans.
Oh, okay.
Yeah, no, we're quite, I mean, I do send to everybody as well in Uppsala and everything. It's how the word spread. Depends. But we don't have any problem filling up the course anyway. There's also people watching the lectures, right, as well. Online. Hello to everyone who watches lectures. A few people.
Mm.
So are you teaching a lot in around, giving lots of lectures?
Lot is, I mean, I present this maybe 3-4 times per year on different courses.
And you have your own courses, of course.
Yeah, and then of course we cover the methods that we support with. So it's the easiest. Tell everybody what you know. Then you don't have to study something new.
Yeah. So I spoke a little bit this year, little introduction about image scanning microscopy. It was very, very small because we have also these.
It will also only be 3 slides here. So
yeah, but I think
and
it's
the
nice.
parameter will just be resolution, resolution, resolution. I assume a perfect sample, perfect contrast, zero background, specific staining, everything is perfect.
Yes. Oh yeah. So, and we speak a lot about autofluorescent mounting medium and
No, there's no autofluorescent in my
sample in the wrong place and things. But you know,
everything is
everything is
specific,
perfect in your
stained.
world.
Yeah, zero background, no autofluorescent.
Yeah, we had this a number of people, including in Ireland actually, who are working with
Yes, there was
emailed you.
no, maybe not Ireland. Yeah, it was somewhere from the islands.
Postdoc with Stefan Hell, who got the Nobel Prize when the super resolution was in the Nobel Prize, and he helped develop the STED super resolution microscopy. So he also manages the advanced light microscopy facility at SciLife in Stockholm. And so he's going to talk to us about the different super resolution techniques. Please, Hans. Thank you.
Okay, yeah, thank you, Sylvie. Yes, as said here, I will talk about super resolution fluorescence microscopy. And there are many different modalities, and I will cover some of these. So, Sylvie already told me that she talked a little bit about this, what I have called matrix here. It's a small array of detectors that works as pinhole. That's one technique that can help you getting a little bit better resolution. Then I will cover what is called SIM, structure illumination microscopy. So instead of just illuminating your sample with the normal wide field illumination, you can use stripes to increase the resolution. And then the technique I worked as a postdoc many, many years ago in Stefan's lab was to do STED microscopy, where you add a little bit of extra laser light to turn off the fluorescence from molecules. And then the last technique I'm going to talk about is this acronym SLMM, single molecule localization microscopy, where you look at individual molecules and you pinpoint their focus.
So
and below here,
just to be sure, the stuff that you called matrix, I called it image scanning microscopy, so that people can connect.
Exactly. And Nikon, we call it NSpark, and Zeiss will call it ARRI, and Olympus Evident, we call it Zora, and then there's a Dutch company that we call it Rescan Microscopy. And there's a lot of acronym there. So also for the single molecule localization microscopy, there's a lot of acronyms. So I hope you at least can get the idea about what the technologies are about, and then the names you can basically forget and make your own names. So below here, I listed a little bit of ballparking resolution areas where you can kind of apply these methods. So of course, going smaller and smaller and smaller is going to also be harder and harder to get the method to work. So there's no free lunch. So let's continue here then. Hello. There. Yes, so let's start with the eye, the human eye as a resolution. You know, you have a pretty good microscope actually inside your head. with a good lens system and actually very, very good detectors. You probably discussed a little bit what can you resolve, and then this thing that you want to resolve, you kind of put that on a detector, and then you have this airy pattern that you have to, so to speak, have separated to say that this is resolved. And in the eye, you can kind of resolve something that is 100 micron, because it's actually a demagnification here of a factor of 10. But luckily, the detector array are around 2 to 4 micrometer sized here. So that means you have a very, very good detector with small detector pixels. And then, of course, in order to improve this, you use the magnifying glass or a sophisticated microscope to boost the resolution here. And if you then look through history, then of course people has been designing microscopes to see smaller and smaller and smaller things. And the revolution was, of course, like 400, 300 years ago, and then all the way up to the late 1800. Actually, was kind of more of a handcraft to figure out better lenses, better illuminations, better detectors, and all like that. And this continues today. But then you know about the Abbe resolution limit, where Abbe kind of coined that, okay, using visible light and the technical instrumentation that you have, you can then get the resolution about basically the wavelength that you use divided by 2. So if we use visible wavelengths 500 divided by 2, then you have the limit of not seeing things that are below like 200 nanometer. But that was, of course, just using one lens and in an illumination scheme that we can today circumvent. One could use many more lenses, for example, or one can use tricks to actually localize things in space and time. So let's take the first step into super resolution microscopy then. Some of you have probably used confocal microscopes, which is a smart imaging modality. You can get optical sectioning by having a pinhole in the detection path. And if you now actually close this pinhole, you can go to higher and higher and higher resolution, actually. And that you can see here schematically, you're closing the pinhole, the resolution may increase, but you also see that the signal also decreases quite a lot. And that's, of course, intuitive. A very, very small pinhole doesn't let through that much light, but it has a very good resolution property. So you can gain like 30-40% resolution by closing the pinhole. So don't be afraid of closing a pinhole on your confocal microscopes when you have a very good staining and strong samples.
I have a question.
Yes.
On your graph here is 0.7, the resolution, and then I guess it plateaus a little bit. So how about closing the pinhole smaller than 0.7? I also heard that 0.7 was some sort of
if you close it too much, then of course you have zero signal. So you have signal to noise ratio of 0, and then of course.
Oh yeah, no, I actually read this graph wrongly. So the pinhole diameter is at the bottom there. Okay, thank you.
Yeah. So closing it too much is of course bad, but close it to like half an area unit. You of course lose at least 50% of the signal, but you gain maybe 20% in resolution. So don't be afraid of tweaking on that one. Yes. So then of course. The first step here was also what Sylvie discussed, this imaging scanning microscopy. So a few years ago, in 1988, Colin Shepard actually figured out if you now, in a confocal microscope, displace the pinhole a little bit, that is kind of making it smaller, because if you have the pinhole perfectly overlap, then the overlap is big, but if it's then slightly skewed, it's kind of smaller, and thereby you can increase the resolution. And then also Colin wrote in the paper that, this super resolution confocal imaging is then easy to do. You just put a lot of smaller pinhole at the side. But now we're talking many, many years ago, the computers to store a lot of images from many pinholes was, of course, not doable. So the computer power was not there. So it took another, 30, almost 40 years to get this going. So today you have detector arrays that are small pinholes, and then schematically here you kind of excite at the center of the sample here, and then you detect at the side shifted, and then you actually need to reconstruct this image and actually putting it back, which is the distance between the excitation and the detection. So you move that in between. So there you can increase the resolution in a confocal microscope by having this array detector. So this image is stolen then from size. And here you see a normal confocal microscope. You actually spend a lot of money to just buy a hole. And then you can have an array detector. On the Nikon system, this is called the N-spark detector. And then you process this image and you can increase about a factor of 1.5 and so on. So it's a quite good technique. Of course, the data is now 30 times bigger than the the top one, so the files are of course bigger. But today we have a lot of nice storage in the computers. Yes, so then I step over to the second method that I was going to cover, structure illumination microscopy. And here you see a schematic of how the principle is here. So if you have a sample which has very, very fine structures here, for pedagogical reason, you actually see these, but imagine now that they were too hard to resolve. So then you can use this trick of illuminating the sample with striped illumination here. And then the illumination pattern and the sample pattern will kind of interfere and generate a pattern like this. And here you see broad line that are separated between each other quite well. So this is easy to resolve because the microscope can actually see these broad lines and everything like that. So this is the image result that is easily picked up by the microscope. But now, of course, we're sitting on an illumination on top of that, but we know this pattern. So we can then use mathematical processing to calculate out and come back then to the unresolved sample. So that's the whole idea about structure illumination, that you're using this stripe to, so to speak, down convert high resolution spatial information that the microscope can collect. And then through processing, you can then crisp up your image and get a factor of 2 better resolution in all different directions here, lateral and axial. And the first person that kind of worked on this was Mats Gustafsson, who moved from Stockholm to California to develop this system. And unfortunately, Mats passed away a few years ago. So we could have maybe shared the Nobel Prize. So here he generated the striped pattern by having a grating illuminating it into the microscope. And there you see different paths of this illumination and interfere. And in this case, it's made the wood pile pattern here. So these are the striped looked on along the optical axis. And then this is a camera-based system. So you have a pretty good field of view, nice detector. So it's one could call this super resolution wide field microscope. And if you then look at the information that is transferred through a microscope, you have an excitation here at a fixed frequency, kind of normal wide field illumination. And then the spectrum goes out in, so this is spatial frequency. That means lines per whatever meter or centimeter or millimeter. So this is structures that we normally don't see. They are too fine to see. But now if we illuminate them with the striped pattern, then of course we have extra illumination frequencies out here that can, so to speak, carry the interference of this spatial structure into the detection of the microscope. Those were those broad lines that you saw in that pattern. And then there's a little bit of mathematical processing to put these frequencies, spatial frequency, back where they belong. And then you can crisp up the image. So here's just one of the first images that Mats took years ago, comparing wide field microscopy and then the SIM microscope. It's a mitochondrial network here. So you also have optical sectioning and good enhanced resolution, a factor of 2 in this case. So in volume, it's 8 times more. And we can support project with this method. So here I just put 2 examples looking at neurons. And there you start seeing this spine structure that sticks out from the dendrite, and the next structure which is about 100 nanometer. And then I don't know if you're looking at bacteria, which are small, then of course you can use this SIM microscope to look at proteins distribution in bacteria, or whatever is your favorite organelle or favorite cell or structures. And then lately, maybe I don't know who's going to talk a little bit about the convolution. Maybe it's Eric that's coming later. If
Yes.
you use a mathematical processing on the image, you can actually crisp up the image, because an image is so to speak a blurred reality through the microscope. So if you can then use a little bit of mathematics to come back to the true sample, then you can actually get a better image. And then here is a processing algorithm, which is called sparse deconvolution. And then you can actually crisp up a SIM resolution down to around 60 nanometers. This is, of course, on perfect samples, beads on surfaces or structure that has 0 background and things like that. But it is doable to gain a little bit more on a SIM microscope. So here is just a fast summary of the SIM. It gives you 2 times better resolution. And then 2 times is actually quite a lot, because if you think you had a microscope that could only measure 2 meters, all people on the planet, except for tall basketball players, will be put in, so to speak, the category of people. But if you can double that one, then you would actually start seeing a whole new species called children. So a factor of 2 can give you quite a lot. It's multicolor, it's camera-based. Field of view is pretty okay. You can at least see a few cells. And then you can use your old labeling, maybe with the antibodies or fluorescent proteins, whatever. It's a little bit slow to fast, depending on how you see it. It's camera-based. So, but the problem is you need to run this stripe pattern on top of it. So each so to speak, collected image consists of about 9 to 15 striped images. So therefore, of course, the speed drops a bit. As in all images, you need good contrast. And here, of course, if you have a noisy image and put processing on it, you can, of course, generate a little bit of artifacts. It is a live cell compatible system. The illumination is weak, camera-based being fast and things like that. Then, of course, live cells cannot move too fast or too much between the imaging there. So it's always the condition of how fast you can image. So that was SIM. So let's now step over to the 2 methods that was jointly awarded the Nobel Prize, where one was called STED microscopy, which Stefan Hell developed, and I worked on in his lab many years ago. And there you actually use laser switching of turning on and off fluorescence. And the other method was called single molecule localization microscopy, where Eric Betzig and W. Murner shared the Nobel Prize, where they actually switched on and off fluorescent proteins to localize where molecules are in the image. So let's start with Stefan's invention then, STED. And they actually shared the Nobel Prize because they figure out that if you can separate molecules in space and time, you can actually increase the resolution of an image. So here Stefan is a young postdoc in Finland, in Turku, where he got the idea. So in a normal microscope, confocal microscope, you generate a focus, and here you have some biomolecules you want to separate. But of course, if these are too close to in space, then of course they will just be merged into the same focus spot. And actually, the focus spot is depicted here to be around 200 nanometer, but then of course it's much worse in the axial direction. But then the idea from Stefan was, let's build a microscope that can do this. A microscope that can turn off molecules in part of this volume, and you only look at one here, and then you move the focus across these to build up the image. And now the question is, how can I build a microscope that can switch molecules off? Well, Stefan, on a Saturday when he was a postdoc there in Turkey, actually read about this in a optics book. And there he was stating something about stimulated emission and fluorescence and spontaneous fluorescence and emission and things like that. So these are the processes you can find in a fluorescence microscope. So we normally send in a little bit of laser light onto our molecules and they absorb this light. So this is actually called stimulated absorption. This is how we excite the fluorophore. And after a while, they relax and then they go down to the ground state, sending out, so to speak, what we call fluorescence, spontaneous emission. And this is slightly redshifted compared to the excitation. So we can use dichroics and filters and things like that to separate the excitation from the signal we want to detect. And that's what you're all doing when you're doing fluorescence microscopy. That's the top graph here. But then in the book that Stefan was reading, it was actually also information from Albert Einstein. And he said over 100 years ago, well, if you can excite the molecule, you can get stimulated absorption. It's also possible to de-excite the molecule by stimulated emission. And that was the process that Stefan then explored to develop STED microscopy. So here at the bottom, after the molecule has been excited, it sits here in its excited state. Then if you shine light that is red tuned here to match the energy difference here between the upper state and the lower state, then this system will be stimulated to emit the same wavelength and photon here. So now you have a switch. If you shine on excited molecules a bit of red light, you can turn them off. But then, of course, if you turn off everything, you don't have any signal. So therefore, you need to build a microscope that has kind of a profile where the intensity is very low in the middle. So here you have a normal excitation focus in 1D. You want to separate all these stars. Well, that's impossible in the wide field or confocal. But instead, you add this laser profile. And now you can, so to speak, stimulate these molecules to go to the ground state, and then you wait for the fluorescence here. You crank up the power on the red beam a lot, and then you can start separating all these stars and increase the factor of about 4 to 5 down here. So that's the whole principle about stimulated emission depletion microscopy. You use an extra laser and the photophysical switching of that onto your system. And any dye can do this, because if it can be excited, it can also be de-excited. Some are, of course, better to use in these systems. So if you then want to build a STED microscope, in principle, you just build your normal confocal microscope, and then you add this laser wavelength and use a little bit of optical element to sculpture your laser into this, what is often called a donut pattern. And then cranking up the power, you can separate on the few molecular level in the image. And then does it work? Well, yeah, it works quite well. I usually show this slide, which was the first user I supported in Stockholm in 2011, 2012. So there was Elena Wikström from Linköping. She wanted to look at a gap junction protein. And here is a confocal image. And there we don't really see the distribution of the gap junction protein. But if we turn on the STED beam, it looks like it's kind of a zipper manner. This protein comes when it attaching cells. And this is, of course, then for a specific protein, IQ gap 1. But of course, with electron microscopy, you can resolve these structural morphological very well even have more resolution than STED, but then of course here you don't have the protein specificity. So the super resolution microscopy method can then bridge a bit down to the electron microscopy resolution. And here is another example. If your favorite organelle is centromeres or centrioles and things like that, then of course these things are also small, few hundred nanometers in diameter here, but then of course these structures are smaller than that of the 2 billion bundles. But then you can actually use STED here to figure out this symmetry and the barrels and things like that. And this is of course then a totally raw super resolution image. It's just with the laser physics that you make the sparsity to work here. But then of course you can use processing on this structure and crisp them up a little bit more. And yeah, whatever your favorite organelle is, or whatever cells or whatever you want to look at, you can then crisp up the image using STED. And this can also actually be done in the axial direction by having a little bit of STED light above and below the focal plane. And there you can look at mitochondria confocally, and now you think your mitochondria are 600 nanometer thick, but that just comes from the optical resolution of your confocal microscope. If you then turn on the STED microscope, you actually see that they are more maybe 200-300 nanometer thick, and you can also see here that outer membrane was labeled. So a short summary of STED microscopy. Well, you can push the resolution maybe a little bit more than the numbers I state, but this is also, I don't want to promise too much in support from the facility, but you get 4 to 5 times better resolution just by turning on the STED laser. It's possible to do this in a multicolor manner. We have different STED lasers, so you can select depletion for green, orange, or red, or you can actually match dye combination that you can use a single STED laser for two dye colors. Then, of course, as you know, in this course, you discussed bleed through and cross excitation and things like that. So that, of course, has to be planned in each project. You can use your own labeling protocols with the primary, secondaries, and things like that, or whatever labeling you have. You can usually just switch to more stable STED dyes in the project. But it also worked with the conventional dyes that you're used to, like Alexa 488 or Alexa 555 or whatever. The field of view will, of course, shrink a little bit here as you understand that if we now have 4 to 5 times better resolution, the pixel size, of course, has to become smaller. Maybe here you scan with a pixel size of about 15 to 20 nanometer. And then, of course, the image would take forever if you would be left with an old field of view, confocally or something like that. So you usually zoom in when you want to gain this extra resolution. So therefore, it can be a little bit fast, but it's, of course, slower with the smaller pixel stepping. You detect a few dye molecules, so you're usually a bit contrast limited then. You need to have low background, and the signal, of course, needs to be specific and strong. It is possible to do live cell imaging. The laser power might be a little bit harmful here, but shifting everything into the far red can help. Then, of course, the things that you want to film live can, of course, not move that much during the images. So, and then now, of course, with this high resolution, maybe 50 nanometer movement or 100 nanometer movement can be bad for you. Okay, that was STED. So let's now step over to the last kind of method that I wanted to present here, which is called single molecule localization microscopy. And here, schematically, you can then imagine that if you would have a single molecule in your focus would, of course, still looks optically like you're used to. You would have a diffraction limited width and a diffraction limited height. But now, by stating that there's only one molecule in this focus, you can then on your camera see a blob, And now you make the assumption, okay, if I can fit this intensity profile and saying that my molecules sits in the middle here, if I have very good signals, I can actually fit this quite well and get the precision value here of the localization, which can then be saved as a coordinate. So my molecules are localized then with maybe 20 nanometer precision here compared to the 200 nanometer precision across here. So by doing this mathematical trick, you can then state that, okay, my molecules that I localized has a higher precision than before. But this demands single molecules. So you can't have more in there, because then, of course, you will not be able to separate. So then the first people that actually made this work to make images in life science was Eric Betzig and his team over in the US. And there they used a system where they excited photoactivable fluorescent proteins with a little bit of UV. So they transferred the reporter here over into a kind of an active form. And this is done then stochastically. So there's a sparse excitation here, and a few of these then turns into excitable molecules. They are excited, read out on the camera. The processing then pinpoints where they are, and then this process is repeated 1 times, 10,000 times, 300,000 times, millions of times. So the first time they did this, it took like 9 hours to make the image. Today we can, of course, do this faster. There are commercial systems that you can use. And here I kind of repeated the experiment that Eric did using photoactivable fluorescent protein. So this is photoactivable GFP. We shine a little bit UV light on it, and here you see molecules blinking. And you can actually kind of get a hint of there is a dendritic tree here. The soma is down here. And here we have labeled the sodium ATPase pump that sits in the membrane of this one. So now we just film this for 20 minutes and collect all these molecules that sit on these structures. And then we pinpoint their center of gravity. And then we can map this into a synthetic image here, and seeing along the dendrite on this spine structure here. In red overlay here is a postsynaptic marker, so you can kind of find the sample, because the sample is totally dark here before you start switching on molecules. And then as you have all the coordinates in the image, you can then start making some statistics here, how many there are there any distribution and clusters and whatever things here. So here you have more of a statistical imaging analysis than before. And there are actually modalities where you can also switch just normal dye molecules. So there was a group at Harvard around Xiaowei Shuang, and also German scientist Marcus and several of his co-workers. They just shine light into the microscope, and then after a while you can, so to speak, get the single molecule regime blinking of molecules just having dye molecules here. And basically how it works, you shine in a lot of light onto the system. It actually turns over to dark states, and a few of them are still in the, so to speak, blinking regime. And then you do the same processing of localizing molecules and to the next frame and to the next frame, and then you synthetically map all those coordinates into an image. And we have supported user with this single molecule localization methods, looking into examples neurons on pre and postsynaptic sites. So this is then basically just an area that are a few hundred nanometers big, and this would just be one blob in a confocal microscope. But now you can detect individual proteins sitting in these synapses. Focal adhesion we also looked at in a confocal image. This would just be a, so to speak, a blurred image. But now with this 20 nanometer precision imaging, you can pinpoint where individual molecule sits. So this is what I call a non-railway in super resolution. So this is synaptic and NEMO complexing, sorting the chromosomes in sex cells. And yeah, you can then start appreciating the extra resolution here, can figure out things. And there in this publication was actually obvious that male sorting of chromosome had a larger spatial distribution that female had. So quite interesting. And then there is actually another method in the single molecule localization microscopy where you use binding kinetics as a trick to get information. So here, if you have three what is called DNA imager strengths, you have an antibody here sitting to a schematic microtubule, and then you have a docking strength here, a complementary docking strength. And you can use this DNA binding kinetics to see when the molecule is on. When it is in free solution, it's kind of, you have a little bit of a background, and then you get an image that can then crisp up the resolution just by binding. So now instead of switching molecules on and off with laser, you here switched on an event by binding, and you can then tune the binding kinetics. And here is an image I borrowed from a French group, Eric Hosey and people in Bordeaux, and they looked also at neurons. And then you can also start counting how many blobs do you see then. So it can be quantitative with the higher resolution. So, summary of single molecule localization microscopy. Well, 7 to 8 times better resolution. You can also get this in 3D. There are some optical tricks that you can actually see that your blink is coming from above or below the focal plane. The field of view, it's again more like a stead size, because if you're going to blink a very large field of view, of course, it's going to take forever. Then, of course, the biggest bottleneck here is probably drift on the nanoscale. One usually use fiducials or some imaging tricks to back process what kind of drift you have in your image, because with this high precision, you really need to control drift. As you understand, it's a little bit slow speed because everything needs to blink, and in order to get the full information from the image, everything has to be appearing at least once. Special labeling, of course, you need to get this photo switching to work or this kinetic binding to work. But in the last 20 years since this field was invented, you today have most of this under good control. Looking at the single molecule, of course, you really need good contrast. That means zero background. If you're an astronomer in the day trying to see stars, it's going to be a little bit of a problem. But if you can decrease the background, you're going to see a lot of blinking stars. Then the models that you're fitting the profile on, they can give different results. But today, after 20 years of method developing, those are also very, very stable and give you good results. So, and that was actually the kind of the general talk I wanted to give you. And this is now a little bit of extra treat. But as you understand, the methods that I can kind of told you now has been developed in the last decades. So of course, new things has happened in the last decades. And this is methods that has just been invented in the last kind of 5-ish years or so plus. So Ralph Jungermann, who actually is behind this, what is called the DNA system here. One calls this DNA paint. You're painting your nanostructure by having a complementary binding with these DNA strength. And now, if you repeat this process onto the same biomolecules, getting this to bind and to blink and to bind and to blink and detected and things like that. And of course, if you repeat this many, many, many times, you can use again this center of gravity fitting approach, saying that, yes, now I can even send in my event to being even more precise here. So if this now was the old precision value of maybe 20, 30 nanometer resolution, Then here you can pinpoint down to this to sub 10 or 5 or even lower resolution. And this method is dubbed RESI. So you enhance the resolution by sequentially image the same thing over and over and over and over again. And then, of course, you realize we really need to have a stable system or a chance to correct drift or follow drift and so we don't get blurry images. And here is one of the results that Ralph published just, I think, half a year ago or something like that. There they wanted to look at glycoproteins and the glycosylation here. And they had put this DNA docking strength on here, and then they used this DNA paint approach and cycle these binding events onto the system. And then they could crisp up the resolution to below 5 nanometers on this, or even down to close to 1 nanometer resolutions. Quite impressive. So now we're talking almost like a factor of 100 than compared to Abbe's resolution limits. So in 150 years, we have come a long way, for instance, in an optical microscope. And then there's another trend and a drive actually by Stefan. So you remember from this STED slides I showed you that you used kind of a donut shape depletion pattern to turn off molecules everywhere except in the focus. So Stefan has continued working on this donut idea, but now using it for localizing single molecules. And then the trend here, or the schematic here shows you that is using the donut to triangulate the position of the fluorophore. And you can do this quite accurately, and with smaller and smaller movement of this donut, you can then, of course, come into finer and finer pattern of where the molecule is. So in principle, you triangulate and detect the signal, and then you have an algorithm figuring out, okay, my molecule seems to be a little bit more to the left of the center here, and then you can then get those coordinates. And here we are exciting the molecules with, so to speak, a reversed profile compared to conventional. So it's a very little laser intensity on the molecule. So therefore, one can see this quite much better than sitting on a high excitation background. And here is just a small movie presenting this, what is dubbed min flux, minimum flux emission. So, and there you use the donut, and then you kind of triangulate around molecules, and then you read out where they are. And here you can then crisp up this to, also about a factor of 100 better than confocal microscopy. And this we also support in the facility with. So all the methods I presented, we support it. If the dye molecule is always on, you can track it by triangulating around it and follow it. And then you can follow molecules, for example, in the plasma membrane pretty fast, because you can move that donut very fast, and you don't need that much signal to say where the molecule is. The last method, which is not a microscope method, optical method, this is a sample preparation method to stretch the sample. So you buy super resolution by stretching the sample. So it's like kind of a balloon, or just pull your skin and you can drag it out, maybe, I don't know how long, but biomolecules can be stretched quite a lot. And here I just show some sample. If you have stretched mitochondria, you can start seeing mitochondria protein with much higher resolution. You stretch centromeres, centrioles, and things like that, and you can start seeing them. Scale bars now, of course, increase, because your cell, instead of the nucleus being 10 micrometer, it's 40 micrometer or so. And of course, the more you stretch, the more resolution you can gain, but the sample also becomes a little bit wobbly, and you need to take care of that. And then, of course, if the volumes increase X, Y, and Z, then of course it's going to take you maybe 64 times longer time to measure the whole volume or whatever. But you can stretch spheroids, you can stretch meat, and I think David presented a little bit on those clearing and expansion when he gave the talk. So you can also try this method of actually stretching your sample, and then of course you can, on top of this, add super resolution optical microscopy. And then this will be kind of the summary slide. In order to plan your project and get successful, of course, you need to select the optical method that you want to use, understand the benefits or the problems with it, so you adapt it to a system where it can actually be beneficial. And then, of course, setting up your project, of course, you need to select fluorophore, how you should label this, because fluorescence microscopy needs something to be fluorescent. And then, of course, you need to preserve your sample in the best way and have low background. And then, of course, in the planning of your project, already ask the question, what am I actually going to evaluate? Because that kind of steers you towards how you should maybe measure things. Would you count things or, yeah, whatever. And then, yes, just think about something that is small. And that's easy because everything is small in a cell. And then this method can maybe help you crisp up your biological questions a little bit better. And hopefully you can, get good results. So that was my talk.
Are you not presenting the facility?
Okay, yeah, there was maybe one slide more. Yeah, I should give commercial for myself. Here we
Yeah,
are.
commercial. Please
Me and
advertise.
Anna. Exactly. So if you need support, just contact me and Anna, and you can go into the SciLife homepage there. So we have a facility there where we nationally support with super resolution microscopy. So that's my job. So,
But
yes.
you're also, if I understand, you're bioimaging, right? So you can also support people from outside Sweden.
Yeah. If you live on the planet Earth, just contact us. And we give you as good support as we can. We know maybe about other networks or even maybe in your country about contacts and things like that. But we support in the Europe Bioimaging Network, in the Bridging Nordic Microscopy Infrastructure Network, in the National Microscopy Infrastructure Network. And so everybody is welcome. But some project, of course, I will send to other facility because maybe you don't need super resolution or you need so much resolution that I need you to maybe investigate electron microscopy or some other modalities. Yeah, maybe I have it on this slide here. No, that was the last one. Oh.
Okay.
Thank
But
you.
it's you have anyway Hans' contact and in Canvas, and if you can easily find him anyway and his facility, and you can contact if you want to know if the techniques that they support will help. And I think that what we can do now is to do so first we're going to take some questions if there are.
Hi, I have a question.
Please.
I think that some super resolution techniques, particularly I know for the single molecule based ones, you need to be very close to the cover slide because the algorithms that are behind, they assume that your sample is touching the cover slide.
Yeah, I mean, historically it came out of turf imaging because it's good to illuminate only a very thin pancake because the volume you're illuminating also creates background. So the smallest background you can kind of have is you go to turf. The excitation intensity also actually becomes higher through this total internal reflection effect. So it's correct. Historically, one usually goes very close to the cover slip. There are possibility to tilt the excitation a little bit into the sample, and then you can maybe go for what is called high-low illumination, and then you can go maybe a few microns. Today, there are a little bit also a trend of trying to go deeper on an oil objective, maybe 2, 3, 4 microns. But of course, as you understand, the more you excite into the cell, the higher background you generate. But it is possible to see a bit into the cell layers. And if you then also go for example, a cleared sample where you lower the background by clearing away background, then you can actually illuminate even in a light sheet microscope and use the light sheet as a excitation profile, and then on the camera seeing blinks. So it is possible actually to go into the depth. And the algorithms that kind of fits the profile, they are kind of all almost assuming that this is kind of Gaussian-like and things like that, even though it's not exactly that. So there are possibilities to go in depth, but of course it will be tricky. I don't know, what samples do you want to look at?
I work with plants, so there is always a cell wall in between what you want to observe.
Okay. Yeah. You can find a couple of publications that has tried super resolution microscopy in plants. I think the biggest problem there is probably the autofluorescence and all the spectra you have from what is in those green suckers.
But if we stick to the root, there is low autofluorescence. There is autofluorescence, but it's better than in the aerial parts because we don't have chlorophyll. But yeah, we have a lot of changes in refraction index because of vacuoles and walls and so on. And we are never touching the cover slip.
No, I can understand that. Of course, with all the refractive index changes, of course, the emission profiles will then, of course, be a little bit distorted. So maybe as you said in the beginning, the fitting algorithm might be not working optimally. But of course, if you can switch all your detection fluorescence into red, far red, which is also where single molecule localization microscopy is usually applied, because there the background is lower, it should be doable to do some kind of imaging with these very, very advanced methods.
Okay, thank you.
More questions? We have actually several STED users this year.
Yeah,
Hi.
that's the hi.
Wait, can you hear me? Yeah.
Yes.
So I have one question, please. So how does the fluorescence lifetime of the fluorophore affect the STED performance? Because I have read that when you have a fluorophore with 1 nanoseconds, it's not ideal for STED. And yeah, I'm not getting perfect STED images, and I don't know if that is an issue, or I don't know, from your own experience, it doesn't affect that much the quality of the images.
Historically, the trend has, of course, been to select fluorescent dye molecules that had a little bit of longer lifetimes, 2, 3 nanoseconds or so. What the STED beam is actually doing is when it's depleting, so the first comes the excitation pulse, exciting every molecule, and then comes the STED depletion pulse shortly after this to de-excite the molecules in the outer rim. And there you actually use the STED laser beam to kind of then instantaneously switch the fluorophore into the ground state, and that shortens the wavelengths. So the STED beam introduces shortening of the wavelengths, and then you wait for the normal spontaneous emission, the fluorescence of the longer lifetime. So that has been kind of a trick to select a little bit longer lifetime dyes, and then you can actually time gate on your detector, saying, okay, only detect events that comes after like 1 nanosecond plus or so, because these would be the real fluorescent signal. Of course, if the STED dye has a very, very short lifetime to start with, then of course you cannot really use that trick. And the other thing is, if something decays very fast, you have to be very fast and getting all the depletion photons there almost instantaneously after the excitation event. So the trend has been that don't select too short fluorescence lifetime on the dyes, because then you can wait a little bit more for the good fluorescence to come out, and you also have a chance that the depletion beams can be effectively for most of the excited molecules. I don't know, did that answer your question?
Yeah, thank you. Yeah, the problem is that I cannot, like, I made my own fluorophores. So then when I try to do the stat with them, like, I'm not getting the best images, and I don't know why. I was thinking that maybe the lifetime, like, it's an issue. But yeah, I'm not getting
Or if it's too fast, then of course you're going to have a lot of photons coming down into, so to speak, what it's the confocal volume, because the stead photons has not hit all the excited molecules. So then, of course, you have to have a shorter stead pulse, but then, of course, shrinking the pulse length and having even more photons in that very, very short pulse will, of course, also introduce bleaching of your sample, or also increasing the background. So it's going to be tricky for you to maybe get it more optimized than what you actually see.
Okay.
So,
Okay. Thank you.
I don't remember. I haven't looked up on some of the shorter wavelength, the shorter lifetime dyes that actually works pretty okay. I don't know what the shortest lifetime is. Maybe they can approach like below 2, 1.5, and things like that. So I think Alexa 647 has a little bit of a shorter lifetime compared to some of the more long-lived stead dyes. So just Google maybe on stead dyes and lifetimes and see the exact explanation what is the best dye and how it should have its photophysical properties is not exactly understood for sure. So it's also a little bit of try and error and be lucky that it works, because the depletion theoretically it only goes down, but there is photons there that can excite the system, so it can go up into higher triplet states and singlet states, and so you're photo switching your whole dye electronic states. They're quite complex. So
Okay, thank you very much.
There are more questions for Hans. Can I have a few more minutes? Okay, but if not, we're going to do this little quiz that we have, which is quite quick. We can just do it here, maybe. And so if you go to Canvas, and I can share my screen. If you go to Canvas and have a look at the quiz that is called Super Resolution Microscopy. So I share here, and Hans can see it as well. Click there. Right, so super resolution microscopy, I'm going to review so you guys can see. Now, we have, so one should turn to super resolution microscopy if the resolution required by the scientific question is between the best resolution of a single point confocal and then a single point confocal can acquire, which is about how much. So you need to write down here, what is the best resolution that a single point confocal can acquire? So think about, the wavelength and things like this. So we're talking about short wavelengths, what would be more or less the best resolution that we can acquire in a single point confocal, and the best resolution that super resolution microscopy can acquire, which is about, and you have to select as well. So you fill this in. And then we'll see, because I think that Hans has updated a little bit yeah, everything moves all the time, so I may have to change the the answers for next year. The limit. Okay. So has anyone, I hope you have finished answering the first two questions. And then in relationship to the parameters listed below, so here are the parameters, in which way structured illumination microscopy and image scanning microscopy, so that's the Airiscan, NSpark, and this type, differ from STED and SMLM, the super duper resolution microscopy. So I tend to call these two mid-range, and this is like super resolution. So field of view, flexibility of the fluorophore. Can you choose the fluorophore you want? The imaging speed, the resolution, the number of molecules that you can label, different colors, the number of channels, and also the imaging depth. So each time you have to select if it's better or worse than STED and SMLM. Then we can look at the answers together. Just raise your hand when you're done. So Hans, if you're, I don't want to hold you there. If you are very welcome to stay and look at the answers with us. But you tell me if you're busy, then I can also send you the answers for your approval later.
Oh, I can stay.
Okay.
My lunch date is at 12, so.
Good. And then, of course, when you submit, you see the answers. Okay, we're slowly our way.
I would, of course, say that the very, very small field of view is the best because then you don't have to waste so much space on the computer. But 2 by 2 is better.
Easier to
And then-
analyze as well.
Exactly. And on the next one, if you can only choose one fluorophore, then
It's
that's
just
much
better.
better. And then you have 5,000 to choose between because then you get lost in space. So,
And if you
image-
only image one fluorophore and 2 by 2 pixels, then you go very fast. It's great.
Exactly. So I think it all depends a little bit here on what you say as the benefit or because
Yes.
everything is complex in microscopy. And then you can prepare very, very small samples too if you only have 2 by 2 pixels. Then you don't have so much background maybe because on the cover slip, the big cover slip, that can be anything on that.
So we have been
And
imaging
imaging
lots of
speed will go up.
yeah, we have been
Super-
imaging lots of samples that were like 1 centimeter
Yeah.
and like half a like several millimeter in thickness and everything. So we're a bit on
Yeah.
the other side of the scale. Not for everybody. So great. I can see now that the majority have answered.
And then the parameter resolution, which I talked about today, that's just one small parameter in microscopy. I mean, there's so much more that is maybe even of more importance. I mean,
So now I want
contrast
to
and
know what Hans would answer so that I can know if the answer I put in are correct
well,
or
I
not.
already told you my answers, and they were
Okay.
totally opposite what's the correct answer is.
So, Hans, what is the best resolution we can image on a single point confocal?
Yeah, in this list, I would select 200.
Okay.
Even though, if of course you could select short wavelengths.
Well, even 200 with blue fluorophore, it's yeah.
Well, even 300 is good. Go
Yeah.
home and explain 300 nanometer to your boyfriend, girlfriend, mother, grandma. What is 200 nanometer? It's small.
It is indeed. And so for super, what is the best resolution that super resolution can acquire? What would you answer here?
Yeah, today, of course, Ralph and Stefan are pushing it down to like below 10, but in our hands and supporting it at the facility, I would not promise anything below 20 nanometer in principle.
Okay, so I put in 20. And then if we look at SIM and image scanning microscopy compared to STED and NIS-Elements, what do you think? Is the field of view worse, better, or it depends?
Yeah, for your stuff, it's of course way worse because you see part of a cell, but of course you have high resolution on that one. So it also
And the
depends on what you.
flexibility of the fluorophores.
Yeah, of course, harder, but also easier because you can maybe only select 2 or 3 that works.
Yeah.
So, but in principle, a bit worse than I would say.
And what about the imaging speed?
Yeah, that's of course for the highest speed, you always go wide field turf camera based.
Yes.
So everything's going to be slower, the more resolution you can kind of get, even though MinFlux is super speedy, but then on a field of view of 2x2 microns.
All right. Okay. So I will go here. What about the resolution then?
Then of course it would be worse if you go for SIM and the
I think
matrix method.
we're answering in the opposite way, so it should be like
Okay,
this,
then
really. Yeah,
then
yeah,
it's
yeah,
better.
yeah.
If
So
you're
this one is
interested in-
worse, yes. And what about the number of channels that we can acquire? Are SIM and ISM better or worse?
Yeah, the higher resolution, the less number of colors you kind of have, but you
Okay.
can do a lot also with
That would
just
be better.
two colors.
Yeah.
You maybe don't need 5 colors or 7 colors because you also need to prepare a perfect 7 colors sample.
Perfect
even
samples, yes. And
But
then the imaging depth.
yeah,
Or-
usually we are kind of surface based. The higher the resolution gets, even though people are of course pushing to go into depth
a
there.
bit deeper. So this would be better. Let's see if Hans' answers are good.
But I have not taken the
Hey,
course.
look at that. Excellent. Very good.
Send me the diploma.
Okay, but so if you guys have so people have raised their hands because of the quiz, but if you have any question that you can ask, you let me know. Julie and Clémence, did you have any question or was it just a leftover hand?
I forgot my hand.
Okay. All right. Well, Hans, thank you very much and congratulations for making it through the quiz. I'm glad that the answers are still actual. So if super resolution improves even more, then I'll have to change the answer to the quiz at some point. So thanks a lot and we'll see you again next year, hopefully. And for us, we are going to go to the workshop